This is module 2.2.3 of the online course on genetic selection from concepts to application to genetic improvement of health. My name is Jack Dekkers from Iowa State University and I will be the presenter for this module, which is about genetic variation and quantitative trait loci. In this module, we will discuss the difference between Mendelian and quantitative or complex traits. We'll talk about the concept of quantitative trait loci or QTL, as well as their effects on phenotype, including dominance and epistasis. Now, to target your learning, I'd like you to keep the following questions in mind as we go through this material. First, is litter size a Mendelian or a quantitative trait? Second, is disease resistance a Mendelian or a quantitative trait? What is a QTL? What is the difference between an additive QTL and a QTL that shows dominance? What is an allele substitution effect, and what is epistasis? We will start by describing the difference between Mendelian traits and quantitative or complex traits. Mendelian traits are the type of traits that Mendel studied in his experiments with peas, for example, pea color, yellow or green, and whether the peas have smooth or wrinkled skin. For these traits, the measured phenotypes are typically discrete or qualitative. In other words, there are only a couple of classes in the recorded phenotypes, for example, yellow and green. Thus, Mendelian traits can be represented and analyzed by counts and frequencies. However, in addition, for a trait to be considered Mendelian, it needs to be controlled by one, or at most a couple of genes, and also not be affected by or only little affected by external or environmental factors. For example, wrinkled peas would be wrinkled regardless of the amount of fertilizer the plant gets. In contrast, quantitative or complex traits are usually measured on a continuous scale, for example weight, and they are typically controlled by a large number of genes as well as by the environment. For these traits, we characterize the trait in a population in terms of a mean and a standard deviation. And the phenotypes in a population typically follow a normal distribution or a distribution that can be transformed to be normal. However, quantitative traits are not necessarily measured on a continuous scale. For example, it is well known that susceptibility to cancer is affected by many genes as well as by the environment. So from that perspective, it is a quantitative or a complex trait. However, the phenotype that is recorded for cancer is typically qualitative. In other words, the individual either has cancer or the individual has no cancer. For these types of qualitative complex traits, we can consider disease susceptibility to be the normally distributed quantitative trait, which is affected by both genetics and environment, but this susceptibility cannot be observed, it's hidden. In these models, the expression of the trait in terms of whether the individual is sick or healthy depends on whether the susceptibility of that individual, which is the sum of genetics and environment, exceeds some imaginary threshold. And the level of this threshold is set such that it results in the observed frequency of the disease in the population. So if the susceptibility of the individual for disease liability is greater than the threshold, the animal gets sick, and if it's less than the threshold, the animal gets healthy. And the threshold is set such that it results in the right proportion of individuals being sick versus healthy. So this so-called threshold model allows us to utilize all the methods of quantitative genetics at the level of the underlying susceptibility, although the observed phenotypes are categorical. So quantitative traits are affected by multiple genes, many genes. They're typically spread across the genome. Now we can find these genes by finding loci that are variable in the population. In other words, loci that are polymorphic, and for which the genotype at that locus is associated with the phenotype for the quantitative trait. For example, if we obtain the genotypes for a genetic marker on a group of pigs, and we find that pigs that have the big Q genotype have a higher average weight than pigs with the big Q little q or the little q genotype, then we conclude that this genetic marker is associated with phenotype for this quantitative trait. We can, however, not conclude that this specific marker, polymorphism, is causative, that it actually causes the change in weight. It's actually more likely that the effect of the phenotype that we see is caused by another polymorphism that is close to the marker that we have genotyped. So these so-called marker-phenotype association studies only enable us to identify regions of the genome that affect the quantitative trait, and we refer to such a region as a quantitative trait locus or a QTL. So a QTL refers to a region of the genome that contains a gene that affects the trait, even though we don't know what that gene is. In the past 2 decades, many studies have been conducted in many livestock species to identify such QTL using genetic markers for many traits. And these QTL findings have been compiled into QTL databases, and my colleague Jim Reese at Iowa State University has been the lead on that project. He is the bioinformatics coordinator of the National Animal Genome Research Program of USDA. For the pig, this database is called the PIG-QTL database. Using this database, we can, for example, get information on all the QTL that have been found in the literature and reported in the literature for growth rate on chromosome 10 of the pig. And you can see that there are several of these QTL, and several of them comprise substantial segments of the chromosome, because these QTL mapping techniques only allow us to find a general region of the chromosome where a gene that affects the trait is located. You can also see that many studies find QTL for growth rate in similar regions of the chromosome, for example here at the beginning of chromosome 10, and they likely reflect the same causative gene. So this is a very powerful resource summarizing QTL information. This graph illustrates how we can quantify the effect of a QTL or of a genetic marker that is associated with a quantitative phenotype. The graph here plots the weight of pigs against their genotype at the QTL or at the marker, for the case we discussed before. Here the red dot represents the mean weight of each genotype, while the normal distribution around each red dot reflects that pigs with a given genotype at that QTL can still vary substantially in their observed body weight. This variation reflects the effect of other QTL across the genome, as well as the effect of environmental factors. Thus, although a pig that has a high weight, for example 115 kilograms, most likely will have the big Q genotype, it could also have the big Q little q. Based on a regression line drawn through the genotype means, we can also define the additive effect of this QTL, which is the average change in phenotype from having one extra copy of the big Q allele. For this example, this is equal to 5 kilograms, because the average phenotype of big Q little q individuals is 5 kilograms greater than the average weight of little q individuals. And big Q individuals also have on average 5 kilograms higher weight than big Q little q individuals. So on average, one copy of the big Q allele increases weight by 5 kilograms. Now in this specific example, the average weight of the heterozygous individuals is exactly at the midpoint of the two homozygotes, and this makes this QTL to be additive. This is, however, not the rule for all QTL. For example, it could be that the heterozygous individuals have the same average weight as the homozygous big Q individuals. In this case, this QTL shows complete dominance, with the dominance effect being equal to +5 in this example. And the dominance effect is the difference between the average phenotype of the heterozygote and the midpoint between the average phenotype of the two homozygotes. Now, this is a positive dominance effect, and positive dominance effects are also the basis of heterosis. In other words, crosses or crossbreds and they tend to be more heterozygous, they tend to have higher phenotypes on average than the parental breeds, and this is referred to as heterosis. Now, in the case of dominance, we also need to reconsider the average allele substitution effect. In this case, going from q to q increases the average phenotype by 10 kilograms, while going from q to q, adding another q allele, does not change the average phenotype at all. Now, if the genotype frequencies in this population are 25% q, 50% q, and 25% q, then each of these changes are equally frequent, so the average allele substitution effect is a half times 10 plus a half times 0, which is equal to plus 5. So that's the same as we had for the additive locus in the previous slide. Now, in practice, allele substitution effects are estimated by regressing the phenotype on the number of q alleles, which is 0, 1, and 2. So the slope of that regression line, weight on the number of q alleles, is equal to plus 5 kilograms for every q allele. So the allele substitution effect, or the average effect of the q allele, is plus 5. However, if the frequency of q is much lower than the frequency of q, then the regression line will be flatter because more of the slope is driven by the difference between q and q than between q and q. In this example, the allele substitution effect drops from plus 5 to plus 2 kilograms per q allele. So the important message here is that allele substitution effects quantify the average effect or the additive effect of alleles at a locus, and these allele substitution effects are dependent on the frequency of alleles in the population. The reason why we focus on allele substitution effects, in other words, the effect of alleles, rather than on the effect of genotypes, is because in breeding programs we are interested in quantifying the value of individuals as parents, and parents pass on alleles. They don't pass on genotypes during meiosis. So during meiosis, gametes are formed that contain a random one of the two alleles that a parent carries at each locus. Then during fertilization, alleles in the paternal gametes are combined with the alleles in the maternal gamete to create the genotypes of the progeny. So before we know which sires and dams are going to be mated to each other, the best way to select those sires and dams is to select animals whose alleles have the highest average values based on the effects of those alleles as quantified by the allele substitution effect. And then the sum of those average allele effects across all loci for an individual is referred to as the breeding value of an individual. Another way to look at this is that allele substitution effects and breeding values refer to the main effects of alleles in an experimental design in which alleles are the treatment factors. So in that context, dominance effects then refer to interactions between alleles at the same locus or the same gene. Now, we can also have interactions between alleles or genotypes at different loci, as illustrated here, and these are referred to as epistatic effects. And they can also be between alleles at loci on different chromosomes. Now, the graphs that I've shown here show examples of two pairs of loci, one without and one with epistasis. The example on the left, we have two loci that show dominance because the average phenotype of the heterozygote is not at the midpoint of that of the two homozygote. So it's not a straight line, it's a broken line. However, there's no epistasis because the effect of genotype at the A locus is the same for every genotype at the B locus. And that's demonstrated by the lines being parallel to each other. So the effect of the A locus on phenotype is the same for the B1B1 as it is for B1B2 as it is for B2B2. However, in the example on the right, there is epistasis because the effect of the A locus differs for individuals that, for example, have the B1, B2 genotype at the B locus than for individuals that are homozygous B1 or homozygous B2. So these lines are not parallel, reflecting epistasis. So this concludes this module, and I want to leave you with the questions that I referred to at the beginning of this module. So is litter size a Mendelian or a quantitative trait? Remember, litter size is measured in terms of counts. Is disease resistance a Mendelian or a quantitative trait if disease resistant is recorded as sick or healthy? Now, what is a QTL? How is it defined? And what is the difference between an additive QTL and a QTL that shows dominance? Related to that, what is an allele substitution effect? And finally, what is epistasis? Thank you for your attention.